So this seems like a lot of slides, but there's not a lot on some of the slides. And some of them are a little repetitious. I think this will take about maybe 2 hours at most. There's also, hopefully you've noticed there's a module for the final review that has this PowerPoint, has a list of the pathogens and a little document about the overall objectives. Okay, so the first slide, which of the following statements is true prokaryotes? So it's important to know the difference between pro and eukaryotes. So do prokaryotes have an endoplasmic reticulum? Do they have a nucleus? Are they larger than eukaryotes? Do they contain specialized organelles? Are some of them multicellular? So they do not have membrane-bound organelles, and they do not contain a nucleus. So that would be B is the statement that's true about prokaryotes. Now for viruses, remember viruses are acellular, obligate intracellular parasites. Their genetic material can be DNA or RNA, but not both. So which of the following does not apply to viruses? They need a host to multiply. That's true. I just said they're obligate intracellular parasites. Are they more complex than prokaryotes? That is not true. They are referred to as parasites. They are not considered living. They're composed of hereditary material surrounded by a protein coat. So B does not apply to viruses. Who's known as the father of bacteriology and protozoology? So if you remember Anton von Leeuwenhoek, he was the first to observe the microbial world. Francisco Redi showed that maggots came from fly eggs. Ignaz Semmelweis argued that surgeons should wash their hands before performing procedures. So it was an early antiseptic technique. Joseph Lister improved upon it by introducing phenol. Back then it was called carbolic acid. They would spray the surgical instruments in the site of the incision. So Louis Pasteur was the father of bacteriology and protozoology. This man developed a vaccine for rabies. His name is also Louis Pasteur. Alexander Fleming discovered penicillin. Paul Ehrlich coined the term chemotherapy. Which of the following statements below supports the theory of spontaneous generation? Well, Louis Pasteur actually disproved the idea of spontaneous generation by doing experiments with the special swan neck tubes. He came up with the germ theory of disease. He felt there were small microorganisms that we could not readily see that caused disease. People thought that maggots just appeared in dead corpses, but they were actually the offspring of flies. So people did have this perception. They could see that living things seemed to arise from non-living things, and that turned out to be a fallacy. Whose experiments finally disproved the theory of spontaneous generation? That was Louis Pasteur. Philip Semmelweis's concept of asepsis in the medical setting consisted primarily of what technique? We mentioned that before, that was hand washing. Which of the following was the first antibiotic to be discovered? It was penicillin, which was secreted by a fungus that was discovered by Alexander Fleming. What's the smallest and most specific level of classification? Well, the biggest is domain. You have prokaryotes, eukaryotes, and the archaea of bacteria. You have phylum, you have family, you have genus, and the smallest classification is species. Which of the following is correct? So this is the correct way to write a species name. You have the genus first and then the species. And it is, you considered the genus should be capitalized, but not the species. It should be in italics or underlined. D is in italics, the genus is capitalized, and the species is not. There's nothing particularly wrong with this. This seems to me a little hard to tell, but it looks also like it's in italics, but it's underlined. But the other choices, A, B, and E, are not correct. Which of these is not a Koch's postulate? So I'll just go over what his postulates are. The first is an agent must be found in all cases of the disease. So A is the first postulate. The disease organism must be isolated in pure culture. So C is also the second postulate. D is inoculation of a healthy, susceptible animal must lead to the same disease. So that's D, the third postulate. And E, or the fourth postulate, the organism must be recovered from the inoculated animal. So B, the organism must be competent. That is not correct. That's not one of his postulates. Competence refers to transformation in bacteria. A competent organism can take up DNA from the environment. Binomial system of nomenclature refers to, and we've already mentioned this, genus and species, but it's also each organism has two names. It's I guess I'm just over explaining it, genus and species. Obviously, genus alone, A, is not binomial. It's bi means two names. Species is just one name. And D, it's two, they have two names there, but it's not the right order. It's genus, then species. What is a prion? Prions are small infectious proteins. There's only one known. It causes mad cow disease, and it is capable of spreading and replicating. As far as I know, there aren't any other infectious proteins known. Properties of light. One of the properties is the bending of light, which is a refraction. Light that gets reflected back from a surface like a mirror. Reflective surface is C. That's another property of light, reflection. Light can also be absorbed by dense materials, dark materials. Can be parafocalization is a property of microscopes and is not a property of light. So resolution is a product of both the condenser and the objective. Remember, the condenser controls the light source, and the objective, the magnification. Strictly defined, resolution is the ability to see two very close objects as separate. Fluorescent microscopy uses blank as a source of illumination. So we talked about several different kinds of microscopes. The light microscope uses a visible light. The electron microscope uses actually electrons, a beam of electrons instead of light. The fluorescent microscope uses UV light, which is just outside the range of visible light, not quite as energetic as electrons. You need something that would fluoresce when the UV light hits it, so you have to use special dyes. Oil immersion. So when you go to your highest magnification on a microscope, you have a problem with refracting light, the bending of light. And so you add a drop of oil and it reduces the refraction, that is A. What's the resolution of a bright field microscope? They don't have the answer, but it's 0.2, I believe it's 0.2 microns. So beyond that, something that is smaller than that, you cannot see the objects as being separate from each other. Which of the following allows visualization, of viruses? Of these microscopes listed here, A, C, D, and E, those are all variations of light microscopes. B is a scanning electron microscope, and you'd have to have a scanning electron microscope in order to see a virus. The ability to see two closely spaced items as separate and distinct units is termed what? The term is resolution. Which of the following is the best for visualizing interior cell structures of living organisms? Actually, the phase contrast microscope and probably the differential interference contrast by Normoski would enable you to see cellular structures in living organisms. The total magnification of an image collected within a compound light microscope is determined by the objective and the condenser, I think is what they're, well, the ocular total magnification, the objective and the ocular. The staining method uses water as a decolorizing agent. So a simple stain is when you use one stain. The Schaefer-Fulton is for like the tuberculosis bacteria that have that, they are not gram-negative or gram-positive. A negative stain, you're actually staining the background. The capsule stain is a specific stain to see the capsules of bacteria, where basically you do sort of like a negative stain and then you use methylene blue to see the microorganism. Uses water as a decolorizing agent is, I'm sorry, I'm not quite sure which one uses water as a decolorizing agent. The B is an acid-fast stain, so there's an acid wash. Sorry, I don't know the answer right off the bat for that. The Zeil-Nielsen acid-fast stain. So I might have misspoke here. The acid-fast stain is to stain for microbacteria. These are the bacteria that cause tuberculosis. They are not gram-negative or gram-positive. They have a cell membrane, a very thin cell wall, and then a thick, fatty outer layer. So this is this one of the most important stains, the gram-negative, gram-positive stain. It differentiates the two major groups of bacteria. And so the first step is you stain with crystal violet, and that's number one. And then you use 4, which is iodine, which will fix the crystal violet stain if it's gram-positive bacteria. Gram-positive bacteria, you remember, have a thick cell wall. Gram-negative bacteria have the lipopolysaccharide layer in addition to a thinner cell wall. So the first step is crystal violet. The next step is iodine. There is then a step in between, an acid wash, an alcohol wash. And then lastly, there's a counter stain. So if they are gram-negative, the crystal violet will wash out, and the saffron will stain them like a pink or a red. So the correct order is C, 1, 4, 3, and 2 is the correct order for the gram stain. Which of the following is mismatched? So acid-fast bacteria, they have that thick lipid layer, mycolic acid. That is correct. The gram stain is the initial step in differentiation of bacteria. That is C. The spore stain stains for the endospores. Remember, some species of the gram-positive bacteria can make endospores. The negative stain is, you can see the shapes of bacteria. So A would be the one that's mismatched. And so this is just generalized matching of different things. Genetic material, number 1. The genetic material in bacteria is DNA. Number 2, a virulence factor. E is a virulence factor in bacteria. It helps protect them against phagocytosis. 3, protein synthesis. In bacteria and in eukaryotes, protein synthesis occurs on the ribosomes. 4, motility. Some bacteria have flagella, which is an appendage of motility. 5, holds the cytoplasm, that is the cell membrane. Bacteria, as well as eukaryotes, are all cellular organisms. They all have a cell membrane and a cytoplasm. Number 6, protects from lysis. This is the cell wall of bacteria. Which of the following is found in eukaryotes but not in prokaryotes? So eukaryotes have a Golgi apparatus. They have an endoplasmic reticulum. They have a nucleus. So the answer is D, all of the above. Remember, prokaryotes do not have membrane-bound organelles. What's the main component of a bacterial cell wall? If you remember, it's those, what we use the abbreviation NAG and NAM. They are called peptoglycans. They are modified sugar, and that's the main component of the cell wall. Gram-negative bacteria have blank as one of the major components of their cell wall. They have the D-lipopolysaccharide layer, LPS. That is one of the major components of the cell wall. The LPS layer also happens to have the endotoxin in it. Which of the following digests peptoglycan? If you remember, it's the enzyme lysozyme, which is also one of our innate immune responses. It's found in tears and in saliva. Ribosomes are the site of protein synthesis D. Remember that occurs in both pro and eukaryotes, so ribosomes are not considered membrane-bound organelle. They are found in the cytoplasm of both prokaryotes and eukaryotes. Bacterial endospores are used for, you could say, bacterial reproduction, but it's really protection from unfavorable conditions. The bacteria becomes dormant. It mainly maintains its genetic material. When it reaches more favorable nutrient and rich environment, it can then reproduce again. And we say it germinates from the spore. Bacteria may use which of the following for locomotion? Cilia are mainly found in eukaryotes. Bacteria have flagella, which is an appendage of mobility. Pili is an attachment appendage. So the answer is D, flagella. Which of the following is common to both pro and eukaryotes? Well, prokaryotes do not have a nucleus. They do not have endoplasmic reticulum. They do not have Golgi apparatus. So what is common to both is they have a cell membrane. Blank protects bacteria cell from phagocytosis and desiccation, which is drying out, and that is the capsule. It is also considered a virulence factor. So if you remember, in metabolism, oxidation is the loss of electrons during a chemical reaction. So oxidation, you remove electrons, you're removing energy, and something gains the electrons. We say it's reduced, it gains energy. Define metabolism. It's the sum of all the metabolic reactions in a cell. That includes catabolism, which is the breaking down of compounds, and anabolism, which is the synthesis of new compounds. Examples of an anabolic reaction is not A. A is a catabolic reaction, the breakdown of sugar. DNA replication is an anabolic reaction because you make a new strand of DNA. In B, you're taking ADP and adding another phosphate to get adenosine triphosphate. So that's would also be considered an anabolic reaction. It gains ADP, gains energy. It is reduced to make a more compound that in turn can release energy. What takes place in pyruvate processing? So if you remember, at the end products of glycolysis, you have two pyruvates. Under fermentation, they can be fermented to regenerate NAD. And aerobic respiration, the pyruvate is further metabolized into the TCA or Krebs cycle, and the products generated there, the NADH and FADH, go on to electron transport to make more ATP. Autotrophs would use which of the following as a carbon source? So auto means self, so carbon dioxide would be used by an autotroph. A heterotroph would use glucose, which is not part of a functional enzyme. So an enzyme has an apoenzyme, which is the protein part, a cofactor, and a coenzyme. The substrate is what the enzyme acts on to carry out a metabolic process. It acts on a substrate and has a site that is specific for the substrate. Reduction refers to the blank during a chemical reaction. So we said that oxidation is the loss of electrons, and the gain of electrons is called reduction. Those are always coupled. We call it a redox reaction, reduction, oxidation. Which is an example of a catabolic reaction. So remember, catabolism is the breakdown of something. So A is the glycolysis, occurs in the cytoplasms, the breakdown of sugar. That is a catabolic reaction. DNA replication is synthesis. B, when ATP loses a phosphate, that would be a catabolic reaction. It is becoming oxidized. It loses some energy. Human pathogens, most human pathogens are chemoheterotrophs, D. Phototrophs and photoheterotrophs are not common pathogens. Chemoautotroph also is not a common pathogen. Phosphorylation refers to the addition of a phosphate molecule, not the removal. You phosphorylate something, you add phosphate. Conversion of phosphate to elemental phosphorus. Phosphorylation is A. Which of the following could be a fermentation product? Well, it's not glucose. So glucose gets first broken down in glycolysis. It gets in products of pyruvate, and under an anaerobic condition, those products can be fermented. And the fermentation product, one example of a fermentation product is alcohol, D. How do anaerobic and aerobic metabolism compare in terms of their net energy? So if you remember, you gain a lot more ATP from aerobic metabolism. I believe you can get as many as 38 ATP from a glucose. Anaerobic, only a couple of ATP. It's not as efficient as aerobic metabolism. Name the name the steps in compounds formed in aerobic and anaerobic cellular respiration. So you should go back and look over that. Aerobic respiration starts with glycolysis, breakdown of glycolysis. Glucose is 6 carbons broken down into 2 3-carbon compounds called pyruvate. Those get then joined with acetyl coenzyme A. And some more energy is generated. The acetyl coenzyme A enters the Krebs cycle, or also called the TCA cycle, to generate more NADH, FADH, which finally goes to electron transport to get more ATP generated. Initially in glucose, some ATP is made directly. That's called substrate level phosphorylation. Some ATP is made during the TCA cycle, but most of the ATP comes from NADH and FADH. Anaerobic metabolism occurs without the presence of oxygen. Remember, electron transport, the ultimate electron donor is oxygen, and the hydrogen ions that are created are then combined with the oxygen to make water. During electron transport processes, there's a buildup of blank on the outside of the bacterial membrane. That is the buildup of protons and electrons. I don't know why they should have a choice saying A and B. It's both protons and electrons. In the initial step of fat metabolism, the fat molecule is broken down into blank by a process called beta oxidation. It's broken down into fatty acids. Amino acids make up proteins. That is not a fat. Glycerol. Fatty acids are joined to glycerol. So actually the answer would be A and C. The fatty acids are remove 2 carbons at a time. Bacteria have blank as their genetic material. Bacteria have DNA as their genetic material. We have DNA as our genetic material. Only some viruses, not all, but some viruses have RNA as their genetic material. How do prokaryotes organize their DNA? They have one single circular chromosome. Eukaryotes have linear chromosomes. Which base is not found in DNA? So DNA has 4 bases, A, T, G, and C. A always binds to T, G always binds to C. T is replaced in RNA by D, uracil. So uracil is not found in DNA, but it is found in RNA. In DNA replication, how does DNA polymerase add nucleotides? It's in the 5' to 3' direction. It is the direction of growth of a new molecule of DNA. Remember, there's a leading strand which grows continuously and a lagging strand because it's growing in the opposite direction. Because the DNA structure is a double helix, one strand is anti-parallel to the other strand. Process of creating a protein from messenger RNA is called translation. So replication just refers to DNA. DNA, each time your cell divides, the DNA is replicated. When RNA is made, it's called transcription. That refers to messenger RNA. It also refers to transfer RNA and ribosomal RNA. Translation is what occurs when the messenger RNA containing the codons and the transfer RNA brings the amino acid. It has the anticodon and the protein is made. And RNA adenine always binds with uracil. Remember, thymine is replaced by uracil in RNA. In RNA, guanine and cytosine bind each other. And that's the same in DNA. That doesn't change. So this is the overall replication of a DNA molecule. And it shows you the leading strand, this lower strand here. So that is growing in a 5 to 3' direction. And the other one is the lagging strand. Let's see if I can move that up, which is synthesized in these what are called Okazaki fragments that are then joined together, where the DNA opens up to start replication. Is called a replication fork. And you can see in bacterial DNA, if you remember, they had a circular chromosome and they get 2 replication forks to synthesize the new, to replicate the DNA. Before the cell splits, bacteria, when they cell splits, it's called binary fission. In eukaryotes, it's called mitosis. So, number 1 is the ligase that joins the Okazaki fragments. 2 is the helicase that unzips the DNA. The enzyme that makes new DNA is a DNA polymerase, makes eDNA. In transcription, the RNA polymerase makes RNA. 5 is the topoisomerase that coils and uncoils the DNA, uncoils it to make new DNA, and then once it's made, coils it back up. The single strand binding proteins, they help to hold the DNA strand open during replication. Which of the following is a start codon? The easiest thing is just to remember it because there's only one start codon, that's AUG. The point mutation refers to the change in blank in a genetic sequence. That's the change of a single base within the genetic sequence. Which of the following can induce mutations? All of these: base analogs, ionizing radiation, which can break the sugar phosphate background, such as X-rays. Ultraviolet radiation causes the adjacent thymines to bind together. So all of these can induce mutations. So promoter region. So is where the RNA polymerase binds to start transcription. So that's called a promoter. The structural genes are the actual genes that code for protein. We do have genes that code for RNA, but structural genes refers to the sequences that code for protein. In the regulation, the operon, if you remember, it has an operator that can bind a repressor protein that can block the RNA polymerase. The RNA polymerase is the enzyme for transcription, and the repressor protein can block transcription by binding to the operator and preventing the RNA polymerase from carrying out transcription. So this just shows you a picture of that. You have a promoter where the RNA polymerase can bind. In between the promoter and the actual structural genes that code for the genes necessary to break down lactose is something called an operator. And repressor proteins can bind to the operator when there's no lactose present. When lactose is present, the repressor proteins bind, they come off the operator. the RNA polymerase can continue down, making these different genes that get synthesized. Lactose gets broken down. When the lactose is broken down, the repressor proteins are freed up and they can go back and bind to the operator, turning off the expression of these genes. Blank is a sequence of closely associated genes that regulate enzyme production. I'm not sure what the term is. You have a series of structural genes that regulate the sequence, regulate enzyme production. I guess they're just referring to the operon, which consists of the promoter, operator, and the structural genes. So some more questions. Otis. So about DNA. So anti-parallel refers to DNA as a double-stranded molecule. When the strands run in opposite directions, it's an anti-parallel structure. So that is 1 matches to E. 2, in DNA replication, you have 1 new strand, 1 old strand. So we say it's semi-conservative for DNA replication. Hydrophobic is something that that repels water. I don't see the answer for that there. Hydrophilic attracts water. Deoxyribose, DNA has a deoxyribose sugar. The molecule is quite simple. It's the sugar, deoxyribose sugar, nitrogenous base, and the phosphate. A binds to T and C binds to G are the nitrogenous bases. Oh, I guess what they mean, the sugar phosphate backbone would be hydrophilic, and the sugar would probably be hydrophobic. I think that's what they were referring to, but deoxyribose is the sugar. So I'm not quite sure what they're getting at there. So rRNA is the ribosomal RNA. It combines with ribosomal proteins to make up ribosomes. The message RNA has the codons that are complementary to the actual genetic code. Number 3, the transfer RNA forms a cloverleaf shape, and it carries the amino acids to the site of translation. DNA replicates in a 5 to 3 prime order. Also, the anticodon is found on transfer RNA. The codon is found on message RNA. DNA has the genetic code. Which pair is mismatched? So these are mutagens of DNA, alkyliding agents. Alkyliding agents have either a methyl or an ethyl group. X-rays can break the sugar phosphate backbone. That's called a translocation. Base analogs are similar to our nitrogenous bases, but they merely mimic them. They can cause mutation. Intercalating agents are like dyes that can insert into the DNA. So they do not remove amine groups. They are intercalation means to insert inside of. So D is mismatched. UV light is causes thymine dimers to occur in DNA. Okay, end products act as an inhibitor, and which type of inhibition? So that would be feedback inhibition. Remember, enzymes can have an allosteric site. And so as it's making products which can build up, they can come back and bind to the allosteric site and inhibit the enzyme. Competitive inhibitions when something blocks the active site, which is where the substrate binds. So end products is feedback inhibition. So if you remember, there's 3 ways that bacteria get genetic variability. They have to get it what we call horizontal transfer of genetic material. And one of those, the simplest, was transformation. And that is the uptake. B, the uptake of free or naked DNA from the environment. So a cell has to be competent in order to do that. Genetic material through a virus is not transformation. So passage of genetic material by direct contact, cell-to-cell contact, that occurs in conjugation through a sex pelus. And transfer by a virus is transduction. A bacterium with the ability to take up DNA from the environment, we've already mentioned, that is referred to as a competent cell. Lysogeny refers to the bacteriophages that infect a bacteria and can transfer genetic material from one bacteria to another. All the conditions below are required for transformation, except, so for transformation, you have to have naked DNA in the environment. The live bacterial cells that take it up must be competent, and a recombination event must occur. Plasmid transfer is not part of transformation. It is part of conjugation, where you have a special plasmid with the genes, what we call fertility plasmid, that has the genetic material to make a conjugation pelus. Now, these terms refer to transduction, which occurs through the infection of a bacteriophage. So in the lytic cycle, the bacteriophage infects the bacteria and causes lysis. And this occurs through a virulent phage. In the lysogenic cycle, we have a temperate phage that does not cause immediate death of the cell. When it incorporates into the bacterial genome, it's called a prophage. That bacteria becomes a lysogen. Viral DNA is inserted into the genetic material under a lysogenic cycle of the bacteria. The viral plus the host DNA is referred to, as we said, a lysogen. Cells capable of conjugation transfer in F plasmid are termed F+. It means they have the fertility plasmid, the genes necessary to make the pelus. Which one of the pairs below is mismatch? Plasmids are circular extrachromosomal DNA. That is correct. HFR refers to high frequency recombination. An F prime plasmid is within the host DNA, and F negative cells are recipient cells. So which one of these is incorrect? So that's has me a little baffled here. These all look correct to me. I might have to look up some of these, maybe change the slides. Which one is not true about the fertility plasmid? So what is true is it has the genes to make the F pelus. It contains genes that control the transfer of DNA C, and it has genes for the autonomous replication of the pelus. B is incorrect. The repressor protein refers to the lac operon, for example. It is not part of this. Which of the following requires a bacteriophage? Well, it's not A, that's transformation. That is the uptake of some naked DNA. Translation is what occurs during protein synthesis. Transduction refers to a bacteriophage infecting bacteria and carrying pieces of DNA, either generalized transduction or specialized transduction. Enzymes that cut bacterial DNA at specific sites, this is our DNA technology, are referred to as endonucleases. The sites at which these enzymes cut the DNA are palindromic sequences. In other words, they can read the same way, either backwards or forwards. That's so the enzyme can recognize that site. The naked virus is one that lacks a capsid. That is false. A naked virus lacks the envelope, which is a cell membrane that the virus takes when it leaves a cell. It takes some of the host cell membrane with it. It inserts its own proteins into it. All viruses have a protein capsid, whether they are DNA or RNA viruses. They have a protein capsid to protect the genetic material. One theory states that prions cause disease by causing normal proteins to fold improperly. And that is A, that is true. Which of the following best describes a capsid? Well, capsids are made of proteins, so it's not A. It is a B, a protein coat that surrounds the viral genome. C refers to the glycoprotein spikes that extend from an envelope virus like the flu virus or the COVID virus. And the capsid is not made of nucleic acids. So B is the correct answer. Teratogenic effects or effects that can damage a growing fetus that can be caused by viral infections of the mother. Can also be caused by chemicals and environmental pollutants. Alcohol consumption is also, and cigarette smoking can also have teratogenic effects. Which of the following are present in all viruses? So all viruses have nucleic acid. It can be either RNA or DNA. They all have a protein coat, a capsid. They do not all have an envelope. Some have an envelope. So the correct answer is D, which is both A and B. They have genetic material and they have a protein capsid. E and F are not correct. Which pair is mismatched? So Griffith was the first to observe transformation. He called what he didn't know exactly what it was. He just knew that he could combine heat killed bacteria, Streptococcus, that had a smooth capsule, had a capsule, made smooth colonies, to the ones that didn't have a capsule, and the progeny all were able to make a capsule. He called that the transforming principle. Hershey and Chase, their experiment helped prove that DNA was the genetic material. Avery, McLeod and McCartney confirmed that DNA was Griffith's transforming principle. Liederberg and Tatum, they studied conjugation, and they also studied transduction and salmonella. So B is the one that's not correct. Viral genes that cause uncontrolled cell division are oncogenic. There are some viruses that can cause cancer. Normal genes on the influence of virus replicate uncontrollably, or, well, let me think here, they are oncogenes. So oncogenes are genes that we have normally. If they replicate uncontrollably, I forget the exact term for viral genes, proto-oncogenes. Scabies, bovine spongiform cephalopathy, Creutzfeldt-Jakob, and Kruger are caused by prions. So remember, prions are those infectious proteins. They are not caused by a virus. But actually, when they first studied this phenomenon, they called them slow-acting viruses because it took a long time before the disease manifested itself. It turned out not to be a virus, but a small protein. Lysol and Clorox are examples of disinfectants. They are not antibiotics, not sterilizing. They could also be considered antiseptic, but usually you wouldn't use antiseptics, are used against living tissue, so properly they are disinfectants. Which of the following agents does not result in sterilization? So ethylene oxide, which is gas sterilization, Tudor's 2% alkaline glutaraldehyde over a long period of time can sterilize. Membrane filters, special filters with very small pores, can filter out bacteria. Boiling is not a sterilization technique. It will kill a lot of bacteria, but it will not destroy the spores. The ultra high temperature pasteurization, I guess can cause, actually, I'm not sure if that can cause sterilization. I don't think so. It certainly reduces the number of bacteria present, but that's not sterilization. Sterilization is the ability to destroy the spores. So the definitions here, cleaning is just the removal of debris. A sterilant is something that that is frees from pathogens. An disinfectant would kill or remove actively growing organisms. A. Sterilization is something that's completely free of life, and an antiseptic is something that's used on tissues. Microorganisms can be preserved by drying them in a frozen state. We call this lyophilization, where you both remove the moisture and freeze them at the same time. That's called lyophilization. An autoclave needs to be maintained at a temperature of 121 degrees Celsius, 15 at a temperature of 120 degrees Celsius, for at least 15 minutes, and the pressure needs to be 15 psi for at least 15. So I'm sorry, that's the first is 121 degrees Celsius, 15 psi for at least 15 minutes, sometimes longer. After taking the same antibiotic during various bacterial infections, patients can become resistant to them. That can be true, that can happen. Antimicrobial resistance could be avoided by which of the following: limitation of indiscriminate use, and compliance, finishing all the antibiotic on the first treatment, not use them discriminately, like a lot of upper respiratory infections are caused by viruses. You don't need an antibiotic. Antagonisms occurs when two or more antibiotics are working against each other's A. Yeah, well, the answer is A. In the Kirby-Bayer test, a microbe that shows a clear halo around a disc containing an antibiotic is said to be resistant, and that is. Come on. Can you call Otis? So a microbe that shows a clear halo around a disc is not resistant, it is susceptible. We mentioned a couple other methods to determine microbial resistance. Sorry, I don't remember them top of my head. You might want to look that section up. Which of the following is mismatched? See if I can remember these. Clindamycin is used for bacterial infections. Grisofulvin, I believe, is used to treat ringworm. Polymyxins disrupt cell membranes, and rifampicin inhibits RNA synthesis. So the mismatched one is D, clindamycin. Which of the following best describes the circumstance under which one would use a narrow spectrum antibiotic? Well, if you knew the identity B of the infectious microbe, and is when you would want a narrow spectrum antibiotic. The three major side effects of antimicrobials. So if you remember, there were three major side effects. One is allergy, an allergic reaction. Another side effect can be toxicity. And the third side effect is they can kill off beneficial microbes that can lead to super infections. A blank antibiotic only affects a small number of microorganisms, so that's a narrow range antibiotic. Broad spectrum antibiotics like tetracycline can affect different groups like gram-negative, gram-positive, for example. Which of the following best describes a parasite? So a parasite is a symbiotic relationship that benefits one, causes harm to another. So E. coli living in our intestine producing vitamin K, it's not a harmful relationship. It's actually commensalism. We benefit from it. A pinworm that infests a human would be a parasitic relationship. We do not benefit from a pinworm infection. And an organism that always kills its host would also be a parasitic relationship, not a beneficial one. A chronic disease is best described by which of the following? Rapid development of symptoms caused by a superinfection. The disease is slow to disappear, caused by two or more pathogens. So a chronic disease is usually our inability to eliminate an infection. C, the disease is slow to disappear. Which of the following is not sterile? Well, cerebral spinal fluid's considered sterile. Blood's considered sterile. They used to think that urine in the bladder was sterile, but it's not completely sterile. Saliva before secretion should be considered sterile. So the answer would be C, but A, B, and D, these fluids should be considered sterile under healthy conditions. So remember, a sign is something that can be observed after examination. The after effects of a disease are B. A symptom is something only a patient can describe to you. And a syndrome would be several of these together. What the patient describes as their symptoms, what the signs are, you observe together, those can equal a syndrome that might help you identify what the person has. The acme refers to when symptoms reach their greatest intensity, B. C, Clostridium tetani, Clostridium botulinum release this heat labile protein. What is it? If you remember, proteins released by bacteria virulence factors are exotoxins. Endotoxins only refers to what's in the LPS layer, gram-negative bacteria. Some other virulence factors the bacteria can release, hyaluronase that can break down the connective tissue, streptokinase, but in particular, the neurotoxin released by the Clostridium is a exotoxin. It is a protein. It is specific. See, it can be easily broken down. Endotoxins are not specific. It's generalized. It is not easily broken down. And pathogenesis, the capability to produce a disease. The degree of pathogenesis is termed virulence. So a cold is pathogenic, flu's pathogenic, COVID's pathogenic, but they have different degrees of virulence. Transmission by a vehicle occurs by a non-living carrier, such as water, air, or food. That would be true of vehicle. Well, that's vehicle transmission. Then you have mechanical transmission. To think about that. A nosocomial infection is acquired in a hospital or other medical facility. That is true. For some reason, I'm drawing a blank on this. Vehicle transmission. Actually, just take a moment. So that is true. Vehicle transmission is transmission through water, air, or food. Sorry, I got stuck on that. Which is the following is an example of a pandemic disease? Cholera spreads worldwide. That is the very definition of a pandemic. The temporary emergence of diphtheria when the Soviet Union broke up, that was not a pandemic. Chicken pox that spreads through a family of 5 children is also not a pandemic. Contracting Shigella on an international cruise ship. It's a infectious bacteria, intestinal bacteria, but that is not a pandemic. But pretty miserable for the people on the boat. So blank is the rate refers to the number of people infected by a disease per 100,000. That would be the morbidity. Cases per 10,000 should be 100,000 people per year. I think that's clearly a typo. The number of people who die is the mortality. When you have an outbreak of disease, the incidence is the number of new infections, and the prevalence is the overall number of people that have been infected. Blank is the organization responsible for control and prevention of infectious disease in the US. That's the CDC, Centers of Disease Control. Transferrin, that is an arin-limiting protein, part of our innate immune system. Blank in tissue become polymorphonucleoleucocytes. Those are the neutrophils. They are first to the site of infection. They are not as strong as macrophages. Macrophages circulate as monocytes in our circulatory system and then become these large phagocytic cells called macrophages. Lysozyme is found, as I said before, in our saliva and tears. It's an enzyme that breaks down the cell wall of bacteria. All of these assist in phagocytosis prevention, except which? So the capsules can protect bacteria from phagocytosis. The acid-fast cell wall of the bacteria that cause tuberculosis. The leukocidin that can inhibit white blood cells. These are all virulence factors. Lysosomal enzymes are not part of phagocytosis, so that would be A. Oh, I'm sorry, I'm misreading. All these that assist in phagocytosis. None of the above actually assist in phagocytosis. I was thinking what can protect them from phagocytosis. None of these. The answer is E, none of these. Opsomerization results in enhancement of phagocytosis. That's true. That is one of the functions of the complement proteins. They're part of our innate immunity. Disease associated with a granuloma formation. In C, syphilis is a granuloma that's formed. Pimples and abscess are not granulomas. Which of the following terms is associated with fever production? Pyrogen is something that causes fever. The hypothalamus regulates your temperature. A pyrogen can cause it to raise your temperature. Interleukin-1 is an endogenous pyrogen that can also cause fever. So our immune system can release factors to cause fever, and bacteria can have factors that can cause us, called pyrogens, that can cause us to have fever. So the answer is D. Histamine is released by mast cells and basophils. So in this case, the answer is C. Histamine is a chemoattractant for phagocytic cells. It also a vasodilator, makes the blood vessels more permeable and wider to accept more blood flow. So these are the older terms used in inflammation. A is cholar, redness. Dolar is pain. Edema is swelling. A tumor, I guess, would also refer to swelling. Rhubarb refers to heat. Hope I got those correct. The difference between classical and alternate pathway for complement. So complement factors do three things. They increase phagocytosis, that's called opsomerization. They can interact to enhance inflammation, and they can allow the complement proteins form a membrane attack complex that will lyse cells in the cell envelope of viruses. So in the classical pathway, C1 starts the process, but in the alternate pathway, there's a series of factors that start it. So the answer is C. Which of the following terms is associated with pus production? That's a good question. Pyrogen is fever, bradykin's pain, prostaglandin pain. So the answer must be D, tumor necrosis factor alpha. One can acquire artificial active immunity by So active immunity means our immune system reacts. Artificial is an outside source. So every time you get a vaccine, that is artificial active immunity. If you've had COVID vaccine or actually some other vaccines, you can get some side effects. That is your immune system. You're not getting the disease, you're getting an immune reaction. And I had it, and it was very uncomfortable, but it was just a side effect of the vaccine. Which of the cells produce memory cells? So if you remember, we said in adaptive immunity, it's the ability of our immune system to remember infection. The only cells that make memory cells are the T cells and B cells. Plasma cells, when B cells are actively making antibodies, they are called plasma cells. So A and B are the cells, the T and B cells are only part of the immune system that can make memory cells. Which of the following is not a characteristic of B cells? Well, B cells do make antibodies. They are found in all lymphoid tissues, and they differentiate from stem cells. They mature in the bone marrow, whereas T cells mature in the thymus. So A is not a characteristic of B cells. An amnestic response is our immune memory. So you get a vaccine, or if you get sick naturally and overcome it, you develop some immune memory of that disease. And it is specific and it is heterogenetic. But it's that response in particular is due to our immune memory. Which of the following is mismatched? So IgE does play a role in allergic response. IgE binds to the mast cells and basophils. IgM is the first antibody produced in response to infection. That's true. IgA is the secretory antibody. It forms a dimer. IgE is a memory antibody. It's part of the memory response. So B is the one that's not the correct match. Blank term, it refers to the ability of our immune system to mount a unique immune response to each antigen it encounters. That is B, specificity. The tuberculin skin test is based on immediate type 1 hypersensitivity due to injection of a purified protein derived from Mycobacterium tuberculosis. That is not true. Type 1 hypersensitivity is the allergic reaction. Persistent or continuous form antigen antibody complex result in immune complex hypersensitivity. That is type 3 hypersensitivity. That is true. DeGeorge syndrome is primarily a T cell deficiency. That is true. Type 1 or immediate type of hypersensitivity is characterized by the precipitation of antigen antibody complex. That is not true. A is not true. B occurs when the body's immune system reacts against its own tissue as if it was foreign. That is not true. C results from prior exposure to an allergen. That is true. D occurs when someone receives the wrong blood type. That is type 2 hypersensitivity. HIV infects and damages CD4 cells. Which of the following are not CD4 cells? CD4 cells are not dendritic cells. They are not macrophages. Cytotoxic T cells are CD8 cells. Helper T cells are the CD4 cells. And that is an acquired immune deficiency due to the HIV infection. Agamma-globulinemia is seen in newborns who do not have B cells. T cells, lacking T cells is DeGeorge syndrome. Which of the characteristics below is false about Clostridium botulinum? So is it a gram-positive rod? That's true. It's a spore former. It can result in food poisoning, it can be an anaerobe. It's not really involved in dysentery. It makes an exotoxin that's a protein that is also a neurotoxin. This gram-negative bacteria causes Lyme disease. It is the, let me think, I think the answer is C. In the module, there is a list of pathogens to know. Shigella is a bacteria that infects the intestine. Trypanosoma is a eukaryotic parasite. Mycobacterium causes, this one D causes leprosy. And the bacteria, one of the bacteria that causes bacterial pneumonia is Klebsiella pneumonia. So I'm pretty sure it's C causes Lyme disease. All the following cause dysentery except, so Entamoeba histolytica is a eukaryotic parasite that causes dysentery. Shigella sonnia is a dysentery bacteria. Clostridium difficile, spore-forming bacteria that can cause dysentery. Pseudomonas aeruginosa is not a dysentery-causing bacteria. It is a gram-negative pathogen. It was actually the bacteria I worked with my master's. Matching, let's see. So rabies, Bacillus zoster is measles, I believe. HPV virus can cause cervical cancer, sexually transmitted disease, warts. Polio, central nervous system paralysis. Did I get this right? One is Varicella zoster. I believe is the measles vaccine. It can also lead to shingles. Let me look up number 2, since I'm having a blank on that. This 2 causes measles. So I need to look up or to keep. Hydrophobia can happen with, with rabies. Polio can cause central nervous system paralysis. Death, rabies can cause hydrophobia. Which of the following organisms is causes malaria? And it is Plasmodium species, I believe the protozoan parasite that causes malaria, transmitted by mosquitoes. Okay, so that is it. I'm going to stop the share there. I'm going to try to proofread that. I think there were a couple of mistakes in that, but most of that information is correct. So you have the PowerPoint, you have a list of the pathogens and what the objectives are. And let me stop the recording.